So as a reminder where we're looking at adaptive immunity, the third and final line of defense. And in order to simplify and clarify the network of immunologic development and interaction, we've broken it down into a series of stages, and each stage we're going through systematically. So we went through lymphocyte development and clonal deletion. We went through presentation of antigen and clonal selection. Now we're looking at the third and fourth stages together. First, we'll look at T cells, and then we'll look at B cells. So stage 3 is the challenging of the B and T lymphocytes by antigens. And stage 4 is when you have the T lymphocyte response and the B lymphocyte response specifically or respectively. So the T lymphocyte response, remember, it is a cell-mediated immunity, whereas in the B lymphocyte response, we're looking at production and activities of antibodies. So we're going to look at T cells first, and then we're going to look at B cells. Now, the T cell reactions are among the most complex and diverse in the immune system, and they involve several subsets of T cells. And the particular actions of those T cells are dictated by the antigen-presenting cells that activate them. So we say that T cells, it's a term we use, we say they're restricted, so they require some type of recognition of self on the presenting cell before they can be activated. This removal of restriction or recognition is provided by the major histocompatibility complex molecule on the antigen-presenting cell. The foreign antigen is complexed with the self MHC molecule, and that is the signal that the T cell needs. So stimulated T cells produce cytokines with a spectrum of biological effects. They are also influenced by cytokines secreted by other cells. The end result of T cell stimulation is the mobilization of other T cells, B cells, ILCs, and phagocytes. Now here you see the characteristics of different subsets of T cell types in the classic T cell response. CD4, so T helper cell 1, and the co-receptor on the T cell is CD4. Its function or its important features, well, it activates the cell-mediated immunity pathway. It secretes tumor necrosis factor and interferon gamma. It is also responsible for delayed hypersensitivity. So this would be an allergy that occurs several hours or days after the contact, and it secretes interleukin-2. T helper cells 2, the co-receptor on the T cell is CD4. These can activate macrophages to expel helminths or protozoans. They can phagocytize extracellular antigens. They contribute to type 1 hypersensitivity, which are your allergic reactions, and they can encourage tumor development. T helper cell 17 or Th17, they have a co-receptor CD4, the same as the Th2 and the Th1. You see all of these, the same cluster of differentiation, which is happening here. And these promote inflammation. Now, the T regulatory cells, so these were all T helpers, okay? Now, T regulatory cell, this is going to have a co-receptor of CD25 in addition to CD4, controls adaptive immune response, prevents autoimmunity, and can contribute to cancer progression. A C cytotoxic cell has a cluster of differentiation 8 as the co-receptor on the T cell. And as far as its functions go, it destroys target foreign cell by lysis. It's important in the destruction of complex microbes, cancer cells, virus-infected cells, plays a role in graft rejection, and requires the major histocompatibility complex 1 for function. Now, a T cell is initially sensitized when you have an antigen MHC complex coming in contact with T cell receptors. Activated T cells then start to divide, and they differentiate into one of the subsets of the effector cells, which we just looked at. So those would be your T helper cells, regulatory, and cytotoxic. Yeah. Now, looking at the T helper cells in a little more detail, the first thing to notice is the central role of a kind of T cell called helper cell. This is the first thing in the response of T cells. There are many different kinds of T helper cells, and they all have the CD4 marker on the surfaces. T helper cells are critical in regulating immune responses to antigens, including those of B cells and other T cells. They are also involved in activating macrophages. This they do directly through receptor contact and indirectly by releasing cytokines like interferon gamma. Some T helper cells secrete interleukin-2, which stimulates the primary growth and activation of many types of T cells, including cytotoxic T cells. Some T helper cells secrete interleukins 4, 5, and 6, which stimulate various activities of B cells. T helper cells are the most prevalent type of T cell in the blood and in the lymphoid organs. They make up about 65% of the population. The severe loss of this class of T cells with CD4 receptors caused by HIV is what largely accounts for the pathology of AIDS. The second kind we're going to look at is cytotoxic T cells, also called TC cells. These are cells that kill other cells. That's their one and only job, to destroy other cells. So there are a few target cells that they can destroy. One is virally infected cells. So cytotoxic cells recognize these because of telltale virus peptides that are expressed on the surface. Cytotoxic defenses are an essential protection against viruses. They also recognize and target cells carrying intracellular bacteria. Cancer cells is a second group that these cytotoxic cells can kill. T cells constantly survey the tissues and immediately attack any abnormal cells they encounter. The importance of this function is demonstrated in the susceptibility of T cell deficient people to cancer. And finally, we have cells from other animals and humans. So cytotoxic cell-mediated immunity is the most important factor in graft rejection. In this case, you see the TC cells attacking the foreign tissues that have been implanted into a recipient's body. Additional T cells, you have regulatory T cells, which act to upregulate and downregulate T and B cell responses. Then there is a subcategory of T cells called gamma delta T cells, which is distinct from other T cells. They have T cell receptors that can be rearranged to recognize a wide range of antigens, but they frequently respond to certain kinds of PAMPs on microorganisms the way white blood cells in the innate system do. So this allows them to respond quicker. However, they still produce memory cells when activated. So because of this, they're kind of considered to be a bridge between the innate and the adaptive immune responses. Then there is a hybrid kind of cell that is part killer cell and part T cell, with T cell receptors for antigens and the ability to release large amounts of cytokines very quickly, leading to cell death. So these cells are called natural killer cells, or NKT cells. Because they have T cell receptors but respond very quickly, they are considered to be another important link between innate and adaptive immunity. Natural killer cells are a kind of lymphocyte related to T cells that act less specifically. They circulate through the spleen, blood, and lungs, and are probably the first killer cells to attack cancer cells and virus-infected cells. They destroy such cells by similar mechanisms as the T cells. And finally, we look at innate lymphoid cells, or ILCs. There are 4 types: ILC 1, 2, 3, and the NK, or the natural killer cells. Scientists have found that the ILCs, the innate lymphoid cells 1, 2, and 3, mirror the actions of T helper cells 1, 2, and 17. And the natural killer cells, the NK cells, mirror the actions of T cytotoxic cells. An important difference, of course, is that the ILCs, the innate lymphoid cells, do not express antigen receptors and do not undergo clonal selection and expansion. So they are implicated in allergy as well as autoimmune diseases also. So here you see a T cell activation happening. There is a reaction. So your antigen has entered with the antigen presenting cell and has activated one or the other cluster of differentiation. Here you see a reaction with a CD4 cell. And then the T helper cell, the CD4 cells, are stimulated by that antigen major histocompatibility complex. So they can differentiate now into a variety of different things. They could become T helper 1, T helper 2, T helper 17, T follicular helper cells, or T regulatory cells, depending on what kind of cytokines the antigen presenting cells have secreted. So a Th1, the T helper cell, here we are, the T helper cell 1 is going to activate phagocytic cells to be better at inducing inflammation. The Th2 cells are going to enhance the antibody response. So one of their important roles is to respond to extracellular microbes, helminths, and allergens. Your TFH, your T follicular helper cells, aid in B cell differentiation. The Th17 cells secrete interleukin 17, which leads to the production of other cytokines that promote inflammation. Inflammation is useful, but when excessive or inappropriate, it can lead to inflammatory diseases like Crohn's disease or psoriasis. So Th17 could be playing a critical role in those conditions. T regulatory cells are broadly in the TH class because they also carry CD4 markers, but they're usually put in their own category. They act to control the inflammatory process, to prevent autoimmunity, and to make sure the immune response doesn't inappropriately target normal biota. So a brief mention before we go on. The T cell system is very complex, and as a summary, T cells are differentiated into 5 different kinds of cells, as well as memory cells. Each kind of T cell contributes to the orchestrated immune response under the influence of a wide range of cytokines. So the research has shown that people who've experienced COVID-19 have fewer T cells, significantly fewer T cells, which leaves them more susceptible to other infections. This is something worth keeping in mind. Now, here we see T cell activation, where you have the antigen entering with the APC, antigen presenting cell, activating CD4 or CD8, like in the previous image. Same thing so far. Now, here you have a CD8 cell instead of a CD4. Just to remind you, here we were looking at CD4, here we're looking at CD8. So for a CD8 cytotoxic to become activated, it has to recognize a foreign peptide complexed with self-MHC1 and mount a direct attack upon the target cell. After activation, the TC cell, the cytotoxic T cell, severely injures the target cell. So this process involves secretion of perforins and granzymes. Perforins are proteins that can punch holes in the membranes of target cells, while granzymes are enzymes that attack the proteins of target cells. The action of perforins causes ions to leak out of target cells and creates a passageway for granzymes to enter. These events are usually followed by targeted cell death through a process called apoptosis. So here you see the process depicted out. Now, the products of B lymphocytes. So far we were looking at the T lymphocytes, which is activated by antigens. At the same time, B cells are being stimulated as well. And here too, there is a series of events that happens. So production of the products of B lymphocytes, you have antibody production happening. And they have different structures, they have different functions, there are different kinds. So a basic immunoglobulin molecule can have various different variable regions. So if we look at this again in more detail, we take one particular antibody type as a model. So let's say there are two functionally distinct segments, which are called fragments. The two arms that bind the antigen are termed antigen binding fragments. Here we go. And the rest of the molecule is the crystallizable fragment because it's the first to be crystallized in pure form. The basic immunoglobulin molecule is a composite of 4 polypeptide chains, a pair of identical heavy chains. Here you see your heavy chains, and a pair of identical light chains. One light chain is bonded to one heavy chain, and the other to the other one. The two heavy chains are bonded to one another with disulfide bonds, which creates a symmetrical Y-shaped arrangement. The end of each FAB fragment, so an FAB is an antigen binding fragment. That's the arm that binds the antigen. So the end of each FAB fragment, which you see here, has the variable region of the heavy and light chains, folds into a groove, which will accommodate one epitope. The presence of a special region at the site of attachment between the FAB and the FC fragments allows for swiveling of the FAB fragments. In this way, they can change their angle to accommodate nearby antigen sites that vary slightly in distance and positions. So these are two depictions of the structure of antibody. This is a diagram, so you see the principal function areas, the FABs, the FCs, and the FABs. FAB with the hypervariable region and the constant non-variable region. This one here is a realistic model of an immunoglobulin. So you see a tertiary and quaternary structure by the additional intrachain and interchain bonds that exist. In the activation of B cells, the first thing that happens is the binding of an antigen. So a pre-committed B cell binds the only epitope that its receptor fits. Antigen-presenting cells can also present antigen to naive B cells. The second step is the antigen processing and presentation. So this antigen is endocytosed by the B cell and then broken down into smaller peptide determinants. The antigen is then bound to the MHC2 receptors on the surface of the B cell. This is what you're seeing going on here. Now, moving on to the third step, which is happening here, you have your B cell, THC cell cooperation and recognition. Sorry, B cell and TH cell, not THC cell. B cell, TH cell cooperation and recognition. For most B cells to become functional, they need to interact with an already activated T helper cell that bears receptors for antigen from the same molecule. This T cell has also been activated by an antigen presenting cell. The two cells engage in linked recognition. So the MHC2 receptor bearing antigen on the B cell binds to the T cell antigen receptor, the CD3 molecule, and the CD4 molecule on the T cell. So here you see this binding happening. Here's your B cell, here's your T cell, T helper cell, and here you see the binding happening. You've got the CD4, you've got the CD3 involved, you've got the MHC2 complex, and you have the processed antigen showing up there. So some antigens are really able to stimulate a strong B cell response without the involvement of T helper cells. Like I said earlier, there are often those repeating units, large polymers of repeating units, like the liposaccharide from E. coli outer membrane or the polysaccharide from the Streptococcus pneumoniae capsule. And they would not need this T helper cell binding and involvement. So they would be skipping these steps here and going directly over to that B cell activation. So in B cell activation, you have a combination of stimuli on the membrane receptors that cause a signal to be transmitted internally to the B cell nucleus, and the B cell gets activated. Now, once it's activated, it gets differentiated into 3 different kinds of advanced B cells. These could be your plasma cells, your memory cells, or your regulatory cells. The clonal expansion is the last step where you're looking at each of these 3 different kinds of cells expanding its population by undergoing mitotic division multiple times. Plasma cells are short-lived. These are the factories that produce the antibodies. They have the same specificity as this original B cell. Memory cells, these are going to be seeding the lymphatic circulation. They will be staying for a long time and ready for encounters with the same antigen. Regulatory cells will be proliferating and then secreting interleukin-10 to regulate the T cell response. A summary of antibody functions. So antibodies coat the surface of a bacterium, preventing its normal function and reproduction in various ways. The second, opsonization. So antibodies, which are called opsonins, stimulate this process that makes the microbes more readily recognized by phagocytes so that they can dispose of them. So I like to say opsonization is optimization of phagocytosis. So opsonization has also been likened to putting handles on a slippery object so the phagocytes can get a better grip. In neutralization over here, antibodies fill the surface receptors on a virus or the active site on a microbial enzyme to prevent it from attaching normally. Here you see aggregation happening or agglutination. This is something you would see if you were doing blood testing or blood typing. So the capacity for antibodies to aggregate or to agglutinate, to clot with antigens, is the consequence of their cross-linking cells or particles into large clumps. Agglutination renders microbes immobile and enhances their phagocytosis. Here you see the lysis of bacterial cells happening. So this is interaction of an antibody with complement can result in the specific rupturing of cells and certain viruses. Here we have an antitoxin being formed. This is a special kind of antibody that neutralizes bacterial exotoxins. The antigen-antibody interactions and the function of the FAB. Now, the site on the antibody where the epitope binds is composed of a hypervariable region whose amino acid content can be extremely varied. The specificity of antigen binding sites for antigens is very similar to enzymes and substrates. Because the specificity of the 2 FAB sites is identical, an Ig molecule can bind epitope on the same cell or on 2 separate cells and thereby link them. So the antigen binding fragment has the possibility to connect 2 different cells. So its main function of an antibody is to immobilize or call attention to or to neutralize the antigen for which it was formed. How does it carry out these different things? We will get to that. So the functions of the Fc fragment. Although this antigen binding fragment will go and will bind the antigen, the Fc fragment has a different binding function. So in most classes of immunoglobulin, the Fc end can bind to receptors on the membranes of cells, such as macrophages, neutrophils, eosinophils, mast cells, basophils, and lymphocyte. The effect of the crystallizable fraction, so the antibody's Fc end binding to a cell, depends upon that cell's role. In the case of opsonization, the attachment of antibody to foreign cells and viruses is followed by the binding of the Fc end to phagocytes. The Fc end of the antibody of allergy, which is IgE, binds to basophils and mast cells, and this causes the release of allergic mediators such as histamine. Different classes of immunoglobulins. So classes, these are different structural as well as functional classes, and they are called isotypes. They're differentiated with shorthand names followed by a letter. So it's Ig for immunoglobulin, and then you have G, A, M, D, and E, 5 different kinds. So IgA is one that is really worth investigating a bit more. The two forms are a monomer that circulates in small amounts in the blood and a dimer that is a significant component of the mucus and serous secretions of the salivary glands, intestine, nasal membrane, breast, lung, and genitourinary tract. The dimer that is called secretory IgA is formed by 2 monomers held together by a J chain. To facilitate the transport of IgA across membranes, a secretory piece is added later. Now, this IgA coats the surface of mucous membranes. It is also suspended in saliva, tears, colostrum, and mucus. It provides the most important adaptive local immunity to enteric, respiratory, and genitourinary pathogens. During lactation, the breast becomes a site for the proliferation of lymphocytes that produce IgA. The very earliest secretion of the breast, a thin yellow milk called colostrum, is very high in IgA. These antibodies form a protective coating in the gastrointestinal tract of a nursing infant that guards against infection by a number of enteric pathogens like Escherichia coli, Salmonella, poliovirus, rotavirus, etc. This is one of the reasons that new mothers are encouraged to nurse the newborns at least for a few weeks to take advantage of the immune properties of this first milk. Protection at this level is especially critical because an infant's own IgA and natural intestinal barriers are not yet well developed. As with immunity in utero, the necessary antibodies will be passed on only if the mother herself has active immunity to the microbe through a prior infection or vaccination. Now here let's look at all the different classes of immunoglobulins. We talked about IgA, which could be a dimer or it could occur as a monomer. IgG we see as a monomer. IgM is a pentamer. IgD monomer and IgE monomer. Now the number of antigen binding sites is highly dependent on this. Here there would be only 2, 2, 2. Here you would end up with 2 times 5, which would give you 10. And here either 2 or 2 times 2, so 4 or 2. Molecular weight varies greatly, as you can see, from 150,000 all the way up to 900,000. Percentage of total antibody in serum, the largest percentage is your IgG. 13 for IgA, 6% for IgM, 1% for IgD, and 0.002% for IgE. Average half-life in serum in days. What this means is that in 23 days, whatever number you had would be reduced to half. So it's kind of a gradual breakdown, a gradual reduction in the number. Not a lifespan, but it's just looking at how long does it take for depletion of the numbers to happen. So here you see a long half-life of 23 days, and then it's reducing 6, 5, 3, 2.5. Does this cross the placenta? Yes. So this one definitely is of value. This should here say fixes complement. So does it fix complement? This is a typo in here. So read this as fixes complement. And again, IgG is a yes. The others are no. Does the FC bind to something? Yes, here it binds to phagocytes, and here it binds to mast cells and basophils. And then when we look at the biological function, here, in the case of IgG, the monomer is produced by plasma cells in a primary response. It is also produced by memory cells responding the second time to a given antigenic stimulus. It's the most prevalent antibody that is circulating throughout the tissue fluids and blood. It neutralizes toxins, opsonizes, and fixes complement. IgA, we see that the monomer is present in small quantities in blood. The dimer, as we talked about, is a secretory antibody on mucous membranes. Remember, this is the one that is important in colostrum. IgM, a pentamer, is produced at first response to an antigen and can also serve as B-cell receptor. However, it has a short half-life. Is the receptor, this IgD is the receptor on B cells and a triggering molecule for B cell activation. And finally, IgE monomer. These are the antibodies of allergy and of worm infection. They also mediate anaphylaxis and asthma. Now, how do we monitor antibody production over time? We look at primary and secondary responses to antigens. So if we really want to look at if we want to study more about it, we can learn a great deal about how the immune system reacts to an antigen just by studying the levels of antibodies in serum over time. So this level is expressed quantitatively as the titer or concentration of antibodies. So here you see a response, a primary response, and then a secondary response to a particular antigen. So the primary response, you've had the first exposure, and this is what happens to the levels. And here you're talking about IgM, IgG, and the total. And then you have a secondary exposure. So when you have the first one, what's happening in this primary response? The early part, this is a latent period, and you have a lack of antibodies for that antigen. But there is a lot going on. There's lots of activity happening. During this latent period, the antigen is being concentrated in lymphoid tissue, and it's being processed by the correct clone of B lymphocytes. As B cells synthesize antibodies, the serum titer increases to a certain plateau and then tapers off to a low level after a few weeks or months. Early in the primary response, most of the antibodies are the IgM type, which is the first class to be secreted by plasma cells. Later, the class of the antibodies, but not their specificity, is switched to IgG or some other class. It could be IgA or IgE. After the initial response, there is no activity, but memory cells of the same specificity are seeded throughout the lymphatic system. Now, when the immune system is exposed again to the same immunogen, could be weeks or months or even years, you have a secondary response. The rate of antibody synthesis, the peak titer, and the length of antibody persistence are greatly increased over the primary response, as you can see from the levels. The speed and intensity seen in this response are attributable to the memory B cells that were formed during the primary response. The secondary response is also called the anamnestic response. The amnesia, remember? So anamnestic is memory. So anamnestic response. The advantage of this response is evident. It provides a quick and a potent strike against subsequent exposures to infectious agents. And we will stop here. I will record the next section starting at that point. I do want to mention one more thing here before we close. So we accept that memory B and T cells are only created from clones that are activated by a specific antigen. So this gives us a much quicker and more effective response in the second exposure and all of the exposures thereafter. But now there is a phenomenon that is being investigated. It's been suspected, and there are some confirming studies. And what it's showing is that exposure to a particular antigen can result in memory cells that will recognize antigens that are chemically related to it, even if those antigens haven't been seen by the host. So this could explain the well-known phenomenon that we've seen in developing countries that vaccines against one disease can provide some protection against others. So in Africa, for example, vaccinating against measles cuts death from pneumonia, sepsis, and diarrhea by 1/3. During COVID-19, during the pandemic, scientists found that live attenuated vaccines, such as those for measles, polio, and tuberculosis, enhance a person's or can enhance a person's innate immunity against other microbes. In low-income countries that struggle to get the COVID-19 vaccines, revaccinating with one of these readily available live attenuated vaccines could be helpful. This realization is actually upsetting the long-held view that memory only exists because of specific exposures. But if we consider how activation of adaptive immunity occurs, it starts to make sense when we consider recognition of epitopes, which are small molecules or small pieces of macromolecules on the surface of microbes. So if other microbes share those chemical signatures, or if they share those epitopes, memory cells are going to react against them as well. And this may lead to the use of non-pathogenic microbes in vaccines against more dangerous ones, which would be a wonderful outcome.